The opening session of the late-breaking clinical trials at TCT had some fascinating antiplatelet studies. One study, the BRIDGE trial, presented by Dominic Angelillo, presented the idea of using intravenous P2Y12 inhibitor, Cangrelor, not an approved agent, as a bridge to CABG in the patients who have received Clopidogrel or Prasugrel, but then have to have that wear off as they await CABG. A neat study where they randomized patients to standard waiting versus using Cangrelor, and they found that they were able to maintain the high level of antiplatelet effect with Cangrelor that then, when shut off 6 hours before CABG, was fully reversed by the time the patient underwent CABG. And they found no statistically significant difference in TIMI major or minor bleeding, thus showing the feasibility and very intriguing efficacy overall and safety of this strategy. So further studies may be needed, but a very interesting approach to a vexing problem that we have constantly. Then another fascinating trial was called Rapid Gene, and this was use of a rapid genetic test for the Clopidogrel metabolism enzyme 2C19 in the liver, where polymorphisms reduce the amount of Clopidogrel that's metabolized and lead to less platelet inhibition. And so they did a strategy of standard treatment with Clopidogrel for all the patients having stenting versus getting the rapid gene test, which is a swab of the buccal mucosa, rapid result, and then they would identify patients with the 2C19 allele, treat those patients with Prasugrel, the stronger antiplatelet agent, and those who had wild type or normal 2C19, they'd give Clopidogrel. And they found that with that strategy, they were able to have no non-responders to Clopidogrel or thienopyridine in that strategy. They found no difference in bleeding overall, and so again, a feasibility and initial testing of this strategy of genetic testing and changing the dosing on the basis of genetic results. So from TCT this year, I'm Chris Cannon for Cardiosource Video News.